Hi, welcome to week 4, and this week we'll be talking about genetics, and this is what I would have done with you in class. So let's take a look. I'm actually going to make 3 videos, so this is the first of the 3. So you read about Mendel this week. He was an Austrian monk who worked with the garden pea plant, as I'm sure you're familiar with now, and he worked out from the crosses that he did that there were dominant traits and there were recessive traits. So here's Mendel with his piece. Kind of interesting. He was a monk before that, or prior to that, he was a mathematician and really a statistician. Um, and so he really applied a lot of logical thought to his crosses because he was assigned to the kitchen part of the monastery and he worked in the garden. So he had full access to the pea plants. So just to give you an idea of what he was looking at here, you can see the flowers, you can see like the male portion of the plant, the stamen consisting of the anther and the filament, and then you can see the carpel, which oftentimes you see that as the center part within the flower, and then up at the top, the stigma, which often is called the sticky stigma, where the pollen will land on there. So you can see in this final one where he was able to cross-pollinate the plants and use like a paintbrush to be able to brush pollen onto the top of the stigma, and then the pollen would grow tubes down to each of the individual eggs or ovum, which would be in each of the ovules. So the offspring for the pea plants that he looked at would actually be the seeds. So sometimes he'd have to grow them up, but he could also just look at what color seeds were being produced too by these types of plants. So to look at some of the other traits that he looked at, the stem length, whether they're tall or short, or sometimes they're referred to as dwarf, the pod shape, whether it's inflated or constricted. So constricted sort of like vacuumed packed sort of, whether they're round or wrinkled. I don't know why, that's my favorite. The yellow versus green. So yellow is a dominant trait to being green for the seed color. Whether the flowers were axial, so around like sort of the stem versus being at the end or terminal. And then purple versus white. And then the pods themselves, green was dominant to yellow. So it's kind of interesting. They could have green pods but yellow seeds. So Mendel set up all of his crosses really in the same way. Let me move myself out of the way a little bit here. So he always started with true breeding plants of opposite characteristics. So looking at tall, or the plant height, so tall versus short, is true breeding, or what we might call purebred or homozygous tall, and then the true breeding short. And that was what he called the P generation. The F1 generation was the product of the crossing of the P generation, and he found that he got all tall. And we would know today that that genotype that those plants would carry would be big T if we're using these symbols for this particular trait. So from this, he saw that one factor showed up over another, that there was one trait that was dominant over the other. So this led to his law of dominance. So he wondered if the recessive trait would reappear in the next generation. So to figure that out, he crossed plants from the F1 generation. Why am I not advancing now? Oh, there we go. So he cross-pollinated 2 plants from the F1 generation, big T little t with big T little t. And he found that in the F2 generation, he had a 3-to-1 ratio every time he did that. So in this particular cross, he would get 75% that were tall and 25% that were short. So he used these crosses to help come up with his law of segregation that says that when 2 alleles for a trait separate, or they separate when gametes are formed. So for example, the big T little t could produce 2 different types of gametes, and each gamete would get 1 factor or 1 allele. So in this case, the big T little t would produce gametes that either had a big T or a little t, because they would split during meiosis. So remember that from last week. And again, we can see this with the cross that he did. Big T with little t little t. Big T can only produce 1 kind of gamete, so that's what's being represented here, and that's what we're going to put on the sides of the Punnett square. So with this cross, we'd have a really small Punnett square with just 1 square with a big T and a little t, and we look at the combination, they can only be tall, and their genotype would only be big T little t. we cross two from this generation, so there could be thousands of plants that were produced here, um, and we'd separate the big T and little t, so big T and little t, because this is representing the meiosis part, the formation of the eggs. And because the other plant or the source of the pollen, or in this case we could say it's like the sperm, would have either a big T or a little t. And then we populate the box, putting together the T's, always putting the dominant allele first, so always being big T, not little t, big T. And we can see as it's shaded in here, all two of these genotypes are going to have the same phenotype. They're all going to be tall, and then the little t would give the phenotype of being short. So we get the 3-to-1 ratio. So Mendel also was able to look at two traits at a time. So the first cross that we looked at was what he called the monohybrid cross. And then he also did a dihybrid cross. And he set this up in the same fashion, where he started with parent generation that was true breeding. So true breeding for both the traits, big T, big R, little r, or sorry, big R. And then short wrinkled seeds. So the two traits we're looking at here are the height of the plant, and then looking at the sort of texture of the seed. So we found also in this case that all the F1 plants showed the dominant trait, so they were tall and had round seeds. To create the F2 generation, he crossed two from the F1 generation. And again, we're just looking at the ratio of phenotypes here, not the genotypes. Found that of the 16 different combinations that were possible, if we set up the Punnett square, 9 out of them were tall and round, which showed both the dominant traits. 3 out of 16 were tall and wrinkled, 1 dominant and 1 recessive trait. 3 out of 16 showed, or were short and round, showed 1 recessive, 1 dominant, and only 1 out of 16, or about 6%, a little bit more than 6% of the time, you'd get ones that showed both the recessive characteristics. So we got this ratio 9:3:3:1 when he did these dihybrid crosses. So this again was in the F2 cross. And we can see here, if we look at, again, just sort of an illustrated version of the cross that he just did. Here it's the parent generation. You can see the one type of gamete each could make. And then you have the F1 generation being heterozygous for both. Heterozygous, again, meaning big T, little t, having one of each allele, big G, little g. And then you can see from that there's 4 different combinations of alleles that can be produced here. And with that, we make a Punnett square that was 4x4 or have 16 boxes. And you can see that's kind of shaded in here. The ones that are yellow were the tall, and ones that had green pods. And then you had some that were tall, 3 that were tall and had yellow pods. And then you had 3 that were short and had green pods, and then only 1 out of 16 were short and had yellow pods. So this led to his law of independent assortment, and this sometimes is a little tricky to understand, but basically for looking at 2 different traits, the genes for those traits will separate randomly when the gametes are formed if they are located on different chromosomes. So I have a little way to help explain this if we look at the next couple slides. So we can show this by putting 2 genes for height and seed shape on the same chromosome, or I'm sorry, on different chromosomes. So here we have 1 pair of homologous chromosomes, and here we have a different pair of homologous chromosomes. So remember, in meiosis, this would be sort of representing our metaphase 1, although I don't have them replicated. I couldn't really figure out how to draw that. So this particular chromosome would carry the dominant allele, and the other homologous chromosome would carry the recessive allele. And then on a different pair of homologous chromosomes, we'd have the gene here for the seed texture, so round versus wrinkled. So I would illustrate this particular, a cell from this particular plant that was heterozygous for both these traits, for being tall and for being round. So tall, big T, and round, big R, little r. So these 2 gene pairs are located on different homologous chromosomes. Each gamete will get 1 allele from each pair, so meaning they'll get 1 big T and 1 T and 1 R in each of the gametes. So the part where independent assortment comes in is how those homologous chromosomes pair up during meiosis is completely random. So in this case, we have them lined up where it just so happens that both the dominant traits are on the left and both the recessive traits are on the right. So if that were the case, So if they're inherited this way, how or whether they inherit a big T or a little t has no bearing on or no influence on whether they're going to get a big R or a little r. So we can see how, and I have them doubled up here so you can see how they're lined up. So big T is on the left, little t on the right, big R on the right, I'm sorry, on the left, little t on the right. So when they split, we would see that, I'm sorry, I don't have them drawn in here, but maybe I can grab my pointer or my pen and see that this would be a big T on this one, and this would be a big R on this one. Okay, and then on this one, these would be little t, oops, and little r. Right, and then this one would be the same here, and these two would be the same. Okay, so we can see that's one possible combination. We could get a big T and a big R in one of the gametes, or we could get a little t, and a little r in a possible gamete. I won't try to draw a circle around them. That's not going to happen. But we can also see that in this setup, the T's have switched places. The chromosomes that are carrying the trait for the height have switched places. So where they're lining up on the metaphase plate. And so in this case, we could get a little t and a big R, or a big T and a little r. So that gives us a total of 4 different combinations that are possible in the gamete. So big T, big R, big T, little t, big R, little t, little r. And this can only happen through independent assortment of homologous chromosomes, and it can only happen if those 2 genes are located on non-homologous chromosomes. So if they're located on different chromosomes. Okay, so I'm going to stop there for right now, and we'll pick up with video 2, looking at sort of going beyond just sort of basic Mendelian genetics in the next video.